Hi everyone, my name is Rachel Dang and I am a staff research associate at the Johnson Comprehensive Cancer Center at UCLA. I'll be presenting our work on ancestry influences on the molecular presentation of tumors. Here is a list of people that have contributed to the work and their affiliations can be found below. To begin, cancer is a disease distinguished by uncontrollable cell division of abnormal cells that have undergone progressive mutations. Epidemiological studies have identified numerous ways in which genetic ancestry, the biological descent of an individual's genetic material, influences cancer presentation and behavior. These influences manifest as disproportionate disease incidence, survival, and mortality rates between ancestry groups. Despite advancements in detection and treatment methods over the years, these differences persist, suggesting that there may be underlying mechanisms by which ancestry acts upon cancer. However, the molecular basis between ancestry and cancer remain unknown. Our work is motivated to identify ancestry-specific trends and explanations for observed clinical and pathological ancestry differences in the somatic mutational landscape. Genetic ancestry can't be changed, but perhaps through this work, ancestry-targeted diagnostics and treatments can be developed and refined to better address disparities in cancer outcomes. To do this, we leverage 3 large publicly available pan-cancer datasets: the Cancer Genome Atlas, the Pan-Cancer Analysis of Whole Genomes, and the Genomic Evidence Neoplasia Information Exchange data, specifically from the Memorial Sloan Kettering Cancer Center. These datasets are abbreviated as TCGA, PCOG, and Gini-MSK, respectively. We use these datasets to conduct a comprehensive analysis evaluating different genomic features such as single nucleotide variant, SNV, density, mutational signatures, copy number alterations, and RNA abundance for ancestry-associated differences. After data preprocessing, a univariate test is applied comparing a genomic feature presentation of different ancestry groups within a given cancer type. Samples of European ancestry act as our reference group to maximize statistical power, as they are the largest ancestry group across all the datasets. For non-significant results after false discovery rate adjustments, analysis ends there. Significant associations after false discovery rate adjustment then have a multivariate model applied to account for confounders such as age and sex, as well as any specific tumor type confounders such as smoking status in lung cancer. Over the course of our analysis, we were able to identify ancestry-associated differences, 2 of which I highlight in this poster. The first is that East Asian ancestry is associated with increased percent of the genome with a copy number alteration, PGA, and positive status for the mutational signature SBS12. SBS12 has unknown etiology but accounts for a small percentage of mutations in liver cancer. Figure 2 presents a summary of the associations we found. Figure 2a is a summary dot map of significant associations found between PGA and various ancestry groups in TCGA. Samples of East Asian ancestry had significant positive fold change in PGA, and if you look to Figure 2b, where individual sample PGA scores were plotted, the median is higher for East Asians compared to the European group in TCGA. Figure 2c is a summary dot map of fold changes for proportion of PCOG tumors that were found to be positive for a given mutational signature for only East Asians. In liver cancer, we see there is a significant positive fold change again, and below in 2D, we see there is a very large difference between the proportion of Europeans and East Asians positive for this signature. We also found that a larger proportion of samples of African descent had more mutations in 4 genes previously identified in lung cancer, including CUX1 and TET2. Additionally, a greater proportion of African samples were also positive for the mutational signature SBS4, which has been attributed to tobacco smoking compared to those of European descent. As with Figure 2, Figure 3 presents associations found. 3a shows a summary of significant differences in the number of samples in a population with an SNV in a particular gene. highlighted in the red box is lung cancer, which has the highest of all the cancers, with significant associations with four genes. 2B then plots the actual data, and you can see visually the number of samples in both populations with and without the mutation, as well as the overall proportion. 3C, similar to 2C, shows the fold change in the proportion of African descent samples positive for a mutation for the mutational signature, and 3D shows the actual proportion. In conclusion, we were able to identify ancestry-associated differences in the somatic mutational landscape. These results are exciting, but there is poor agreement between datasets. For example, if you look to the East Asian ancestry results, you can see that there is a significant multivariate Q value for TCGA in lung and liver cancer, but we don't see the same in PCOG. These results would be much more compelling if we saw an agreement between them. We attribute disagreements to the drastically different sample sizes between data sets and for the ancestry groups within them. Geographical differences in sampling also play a role here, as do the differences in molecular profiling techniques. Ancestry-associated genomic events are complex, multifactorial, and interacting. This warrants a further thorough study. To fully illustrate ancestry effects in cancer, we must consider correlated factors and attempt to control for them by sampling more diversely with greater emphasis on complete annotation to better serve a wider range of patients, no matter their background.